computer. All right. So thanks also for the invitation, Ting, to speak to you here today. So indeed, my lab is in chemistry. Actually, my students are a mix of chemists, microbiologists, and also biochemists. And today I will focus more or less solely on those projects in our lab that I think you could classify as bioengineering. And so, as Ting said, in the last 10 years we've become really interested in natural product biosynthesis. At heart we are enzymologists, though, to try and understand enzymes, how they work, and then hopefully use that understanding to manipulate enzymes and make them do things that we want to do, and that's where the bioengineering comes in. And so I'll again focus on enzymes today that have both practical applications as well as being interest to a chemist / biochemist like myself. And so I decided to talk on one project in the lab that focuses on RIPS. Chances are high that you have no idea and never heard about RIPS. It's an acronym that has just been introduced this year, and that hopefully will catch on. And it's on a class of molecules that turned out to be the 5th largest class of natural products that are out there on Earth. But only the genome sequencing efforts of the last 5 years or so have illustrated that there is this large group of molecules that are made by a common biosynthetic pathway, and I'll illustrate what that biosynthetic pathway is and why it is so uniquely suited to bioengineering. So first, a little bit of a justification, I guess. This is a cover from Chemical and Engineering News, a magazine from the American Chemical Society. But you could also go to other magazines like Time magazines or People magazines and find covers where people point out the problem of antimicrobial resistance to our current arsenal of antibiotics. And this was in the year 2000. You cannot really see that, but it's 13 years old. This is in the year 2008. And so we've known really for 2 decades that this is a problem. We realized that this problem is only increasing in severity. But unfortunately, as a community, we're not really doing anything to address the problem. If anything, it's getting worse. So you can see here, as the bacteria are increasingly resistant, whether we're talking about Staph aureus or Enterococci or Pseudomonas, these lines are not going to change. They're only going to go further up. But the number of antibiotics that has been approved each year by the FDA is essentially a linear line down. And there is no sight that this is going to turn up anytime soon. As a matter of fact, while in 1990 there were 18 companies that were working actively on discovering new antibiotics, by 2011 that has declined to just 4 companies, large companies. And so I think most of you know why that is. If you have to invest a certain amount of money to develop a new drug, it turns out that that amount of money is always about the same, no matter what kind of indication you develop a drug for. The timeline is always about 10, 12 years. The amount of money that it costs is about $1.2 billion, as it has been estimated most recently. But your return on your investment is very, very different. If you develop a new drug against something that you take the drug for 2 weeks, which is true for most antibiotics, the return on investment is actually very light. Whereas if you put that same amount of money in an indication that is a chronic disease or a lifestyle disease, diabetes, heart disease, you name it, now your patient is going to take your pill for a very long time, maybe even till they die. And as a result, it's a very simple equation and a very simple solution for the pharmaceutical companies. There is no money to be made, only money to be lost to invest it in developing new antimicrobial agents. So we have this problem, but it's not being addressed unless society, hence governments, are going to put money in trying to find a solution. Over time, I think the financial situation will change. If enough incurable infections start occurring, then of course now there is going to be a more incentive financially to start addressing it. But then if it takes 12 years to find a new antibiotic, we're far away from actually solving this problem. So I have no thoughts that we as a small academic labs are going to solve this problem either. But I want to point out that this is the economic problem, but there's also a scientific problem. And that is when you realize that about 75% of our current antibiotics are derived from natural products. So compounds that are made in nature to kill bacteria. And so unfortunately, together with this declining line here, there has also been a decline in natural product research. So even though we know that 75% of all our antibiotics are natural product derived, 50% of our anti-tumor drugs currently on the market are natural product derived. All but 2 large pharmaceutical companies have closed down their natural product discovery platforms. And the reason for that, as I said, is a more scientific reason. And that is, as has been nicely illustrated in some reviews by Dick Balls at Cubis Pharmaceuticals, the fact that some molecules are very prevalent in the environment. And if you ask how were our current antibiotics discovered, almost all were discovered by taking soil samples from all over the world or culture collections from all over the world and simply looking in a phenotypic assay, is there something in this scoop of dirt outside your lab here that kills a pathogen? Or is there something in a scoop of dirt from Hawaii, China, Europe, you name it? And that's how we found natural products. That also means that you find the ones that are more prevalent first. That also means that those molecules that are present in high frequencies form a smoke screen to find the molecules that are present less ubiquitously. So at Cubis Pharmaceuticals, they have estimated that to find daptomycin, which is a molecule that was the most recent introduced natural product from Cubis Pharmaceuticals, that they had to screen about 10 million samples. While you're screening 10 million samples to find your next antibiotic, about 1 million times you find something else that gives you activity, but only after a few steps of finding out what is that material that gives you activity, you find out you have essentially rediscovered this again. So the rediscovery rate at Cubis Pharmaceuticals, and this has also been detailed from other pharmaceutical companies, was higher than 99.5%. And as you're a pharmaceutical company, even though for anti-tumor compounds, you don't have this financial or almost disincentive to develop a new anti-tumor compound, it still means that you have to spend way too much money to find the molecules that are out here in this regime. And hence they have closed down these platforms. So I'll call that the discovery challenge. Of course, then there's another challenge for natural products. That's the structural complexity challenge. That even if you find a molecule that has great properties, it kills whatever you want to kill, most often it doesn't have the perfect properties to be used in a human. And so typically what the pharmaceutical companies do then, they do medicinal chemistry. They tinker with the molecule. But if you have a molecule that is really complex, tinkering with that molecule, changing a methyl group here or a hydroxyl group there, again becomes very difficult and very expensive. And that's another reason why natural products have fallen out of disfavor in the pharmaceutical companies. And so all of this doesn't look very good with respect to natural products as our future drug leads for whatever indication. Now, fortunately, there's one thing that has happened over the last decade that make many people suggest that natural products could become a great source of leads once again. And that's, of course, the amazing advances in DNA sequencing, such that it's now very cheap and very quick to sequence genomes. And so right now, if you go to the website, there's about 15,000 bacterial genomes, either completed or partially completed. And something that has come out of all of that sequence information is that organisms can make many, many, many more molecules than what we see if you grow that organism in the lab. When you grow it in the lab, you grow it in a defined medium, and that organism, if you're lucky, makes one compound, maybe 2 compounds. Whereas in the complex environment where the organism lives, it gets certain stimuli from other organisms. And so it can make up to 10 to 40-fold more different molecules that we have never seen. We don't know their structures, we don't know their activities. So now we can reverse it then. Rather than looking for the molecule as it has been done for almost 8 decades, you can first find the genes, and then with the genes start finding the molecules. The big advantage of that is that you can see everything that is present in frequencies that you otherwise would never detect or never find. And the second real good advance, or I would say advantage of this method, is that you immediately see from the genes that this is a molecule that we know about and that we don't want to follow up. So we and other laboratories are trying to develop methods to take gene clusters that have all of the genomic information in order to make new molecules and put them into friendly hosts to see if those hosts will be able to make the new molecules. So we do two things. We do genome mining genomes for new natural products. I won't really say much about that today at all. The other thing we do is then try to engineer the pathways to see if we can make analogs addressing this structural complexity challenge, not by using synthetic chemistry, but by using bioengineering, and that's what I will focus on most of today's talk. So the question then becomes, what are you going to work on? 15,000 genomes, say that there's 40 different molecules that can be made per genome. You get an incredible number, and that number is only going to increase. So we've decided to look for two classes of molecules. One are ribosomally synthesized and post-translationally modified peptides, and that's what this talk will be about. Another class that we work on in the IGB in collaboration with Bill Metcalf in microbiology and also Huimin Zhao in chemical engineering and Satish Nair in biochemistry focuses on molecules that have phosphorus carbon bonds. I'm not going to say anything about that, but I do want to point out that that's another area of research, whereas these are very large molecules. And so here we really need the enzymes to make them. These are, of course, very small molecules, and here we can use synthetic chemistry. So here we mostly look for discovery. Here we look for discovery, but also bioengineering of the pathways. So this niacin molecule has been used for more than 40 years now in the food industry. As a matter of fact, if you talk to people at Kraft here on the west side of town, they add this stuff to almost everything they make. And so you have definitely ingested that, even though you probably don't know it. It's naturally present in milk. And despite having been used for 40 years to combat foodborne pathogens like Clostridium and Listeria, no big resistance problem has developed over time. And so this has been a really phenomenal molecule. Ting certainly knows about this molecule as well. And so you might say, why am I not taking this then when I have an infection? And the reason for that is that this went into clinical trials, and it did very well. It was 16-fold better against vancomycin than vancomycin against MRSA, methicillin-resistant Staph aureus. But unfortunately, this is broken down in your blood too fast. So we need to change the structure of this molecule if we want to return it to potential therapeutic applications. And so synthesis is just too expensive of this molecule, and so that has never been tried. But now, with bioengineering methods, we can actually try and achieve that. And I'll tell you a little bit about how we try to do that. So first, how are these molecules made? So this gets me to these RIPS, these ribosomally synthesized peptides that are then post-translationally modified. So it turns out that this goes on in nature in all domains of life in an incredibly high amount, but we've only recently learned about that. So you have a peptide that is encoded on the DNA, transcribed to the RNA, and then translated to a linear peptide. That linear peptide is much larger than the final product. A number of enzymes are going to act on that linear peptide only in the core region. And once these enzymes have done what they are going to do, which is introducing a lot of functionality, then the leader peptide in the recognition sequences are going to be removed by proteolysis, and what is left is your natural product. Okay? And so it turns out that more than 30 different classes of natural products are made by this type of pathway. And one example is, again, that molecule niacin, where the post-translation modifications are installation of these rings and these double bonds here that hopefully you can see. So in retrospect, it's no surprise that nature uses this so frequently, because there's some really attractive aspects. If you're an organism and you are making a natural product, this is the most evolvable way to make a natural product. So any non-silent mutation in the DNA leads to a new compound, as long as the enzymes tolerate that non-silent mutation in your core peptide. And that is the other amazing thing about how this has evolved, is that these enzymes are naturally promiscuous. So they are intrinsically promiscuous because usually they recognize the leader peptides in the recognition sequences to do their chemistry on the core peptide. So most enzymes recognize their substrate where they do the chemistry. These enzymes, they do recognize to some extent the core peptide, but in almost all cases that people looked at, it's the leader peptide that really turns these enzymes on, if you wish. And what that allows nature to do is make these core peptides hypervariable and make all kinds of different molecules with one or two enzymes. And so, of course, these are also comparatively short pathways. It's usually maybe between 3 and 7 or 9 enzymes. And all of these nice, attractive aspects for the producing organism are, of course, also attractive aspects for bioengineering. Short pathways, great. Ribosomal, we don't have to worry about any building blocks. These building blocks are all there. The amino acids are there. Highly promiscuous enzymes, great. Highly evolvable, we all need to do is to make mutations. So we've been looking at these for some time to see what can be done and what cannot be done. And now they're easy to find in genomes as well. So I'm showing here 5 of those 30 different classes of molecules just to give you a flavor, to show you that there's all kinds of different chemical structures that are introduced by these post-translation modifications. These are not your canonical phosphorylations and glycosylations and sumulations and other things that happen in mammals. These are just all kinds of weird chemical structures that are introduced into the peptides. All of these, as you can see, almost all of them are cyclic. There's no coincidence there. They're cyclic to provide organization to recognize their targets. You prepay in a way the entropy penalty. And furthermore, by making peptides cyclic, you really prevent any degradation by exoproteases, so either amino or carboxypeptidases. And so these are much more stable than your typical peptide. They're made in all domains of life: bacteria, cyanobacteria, higher organisms, fungi. They also make in archaea. And so this is a solution that nature has developed that is really in all domains of life. They also target all kinds of different processes, whether it's enzymes, whether it is scaffolding proteins, small molecules, you name it. So this is that term RIPS. So the community that works on these molecules has come up with this name to illustrate to the larger communities that this is a really large class of molecules, and to kind of like get the word out that RIPS is something that we perhaps should pay more attention to than people have paid attention to. Although in most cases, for instance, this was not known to be a ribosomally synthesized peptide. That was not known. This was. That was not known. That was not known. All of that has come out of genome sequencing. And so it's a fairly recent discovery. So let's go back to nisin then. How is that made from a linear peptide? And it's really remarkably efficient, the pathway that nature has chosen. You need to do just two reactions on a linear peptide. And those 2 reactions are very simple reactions to a chemist. It's simple elimination of water from serine or threonine. That results in a dihydroalanine or a dihydrobutyrene, and you can still see there's some of those structures in the final product. Now this is electrophilic, so reactive at this carbon here and there. And you have good nucleophiles in peptides that are cysteines. And hence these cysteines are going to attack here. They're going to first give us an intermediate that's called an enolate. And then in almost all cases, the protonation of that enolate results in inversion of stereochemistry from the L-serine and L-threonine that we started with. That's again important because that makes these compounds even more resistant to proteases. Proteases don't like to act on peptides that contain these stereocenters. Now we need to make 5 of these rings, and I will use this shorthand notation for these lenthionines that have given rise to the name lentipeptides or lentibiotics if they kill other organisms, and methyllenthionine if they come from threonine. So we need to make 5 of those rings. And again, the amazing thing is that there's a single enzyme that comes in and it can dehydrate 8 different serines and threonines that are in very different sequence environments. And it can do that because it's the leader peptide that gives this dehydratase its promiscuity. So it can do all of these dehydrations. It breaks 16 chemical bonds. As you notice, none of the serines and threonines in the leader peptide are affected whatsoever. Then the second enzyme comes in, and it has a really challenging task because it has 5 cysteines and 8 alkenes to react with. And so if you just do the math, there is more than 6,500 different ring topologies that you can make. And if you can also bring in stereochemistry, you can make, I think, more than 5 million different molecules. But the enzyme NIS-C makes only a single molecule, and that is shown here. It makes sure that these cysteines all react with the right partner, and that the right stereochemistry is put in. And so note that we form all kinds of different rings. Here's a 4 amino acid ring, here is a 7 amino acid ring, here we have overlapping rings. Some reactions occur with dihydroalanines, others react with dihydrobutyrenes. So it's kind of like hard to imagine how this enzyme is controlling that. And we've never seen any incorrectly cyclized products, at least for this particular enzyme. So once that has taken place, a protease comes in. It's a dedicated protease. It removes the leader peptide that has done its job, and you have nisin. And so, we have a pathway where we have just 2 enzymes that we have to worry about. We have been working for quite a while now to see if we can make nisin analogs using these enzymes. So we have a number of different directions that we do. We try to understand how these enzymes can do this phenomenal chemistry, because we are enzymologists. We have tried to make analogs of nisin, and there's of course one very easy way to do that, by making mutations by site-directed mutagenesis. Today I'll tell you about going beyond that, because then you're still limited to 20 natural amino acids, and we don't want to be limited to 20 natural amino acids, because we know a little bit about what goes wrong with nisin in blood. And we think one of the things that goes wrong is you get hydrolysis here. We don't think that you can actually solve that problem with any of the natural amino acids. So we would like to bring in non-natural amino acids in order to keep a double bond there, but no longer have that hydrolysis take place. We've also done, and I won't tell you about it, put non-antibiotic peptides after that leader peptides. You make all kinds of different rings. And to some extent, the answer to that is yes. There are some limitations, but you can make libraries of cyclic peptides using these type of enzymes. So I'll focus today on a little bit what we know about these enzymes, but not too much. But how can we put other structures, how can we put non-natural structures into a molecule like nisin? Now let me first point out it's not just nisin that is of interest to biotech. Nisin, as I said, has been used commercially for a long time. It's about to be approved also for the treatment of bovine mastitis. Here is a molecule that is under development at a company called Nikons. It's called microbisporsin. And I'd like to show this one because you can see that in addition to the rings, and we believe that these rings are there to get a scaffold that can recognize a target, that there are additional post-translational modifications. For instance, a chlorination of a tryptophan, a hydroxylation of a proline, a decarboxylation, as well as dehydrogenation of a C-terminal cysteine. We believe, although we don't have really good evidence for it, but we believe that these additional tinkerings to the molecule are essentially to improve further the target binding and also the stability of these molecules. And I'm about to show you that that's essentially what we do also by bringing in synthetic chemistry and merging that with those enzymes. Another molecule is called duramycin. This binds to phosphatidylethanolamin, and as a result it's in phase 2 clinical trials against cystic fibrosis. This molecule, actagardin, is under development at Novactor Biosystems for the treatment of Clostridium difficile. And this molecule actually has very weak antimicrobial activity, but very potent activity against neuropathic pain in mice models. And so Sanofi Aventis is developing this as a potential anti-allodynic molecule. So I'm going to switch to another molecule called lactosin 481. And the simple reason I'm going to make that switch is that this molecule is the one that we were first able to make in the test tube with enzymes that we had purified. And hence we know the most about it, and we also have done the most with it with respect to engineering. The story I'm about to tell, we're also applying to other molecules that actually, quite frankly, might have more interesting profiles than lactosin 481 has, which is not the best antibiotic out there. Its biosynthesis is very similar. Again, a dehydration step, and that is then followed by a cyclization step. The big difference here with nisin that I showed you previously is that it's one and the same enzyme that does both steps, and that operationally actually simplifies for us to do the engineering. So we want to bring in non-natural amino acids For that we have to use synthesis, but we don't want to synthesize the whole peptide, which is 50 amino acids, and the yield of that would be really rather poor. So what we do instead, in the latest rendition of an initial platform that was developed by a former student called Matt Levengood, and that we published some time ago, the improved version, we express the leader peptide in E. coli. We do a little trick such that we can attach an alkyne. And then on a combinatorial peptide synthesizer, we synthesize the core peptide. And now, because this is synthetic, we can bring in, and there are literally thousands of different non-natural amino acids that you can purchase. Or if you have specific structures in mind, you can synthesize them yourself. Then we're going to bring these two segments together in a reaction that chemists call click chemistry. So it's a very, very useful reaction that can be done in aqueous environment in the context of biomacromolecules. And that then gives us something that has still the leader peptide and analogs of the core peptide, but they're now linked by something that is non-peptidic. So we didn't really know if the enzyme was going to tolerate that, but it turns out that the enzyme, in at least 2 different enzymes that we have tested thus far, tolerates this to then do still all of the chemistry. And then we can remove this simply by using a commercial protease, in this case endoproteinase lyc, which cleaves here, and that removes the leader peptide and this thing that we put in between. And so using this platform, we have looked at a large number of non-natural structures. I'm just going to skip through a few of them. You can put in threonine analogs. These are all dehydrated. You can put in peptoids. These are structures that are well known to further decrease proteolytic susceptibility. So what you do in a peptide, you take the side chain that is normally attached to the alpha carbon, you put it on the nitrogen. And so this would be a cysteine analog, this is an alanine analog, and so on. This is a leucine analog. These are also tolerated. You can put in beta amino acids for the same reason. So we put now an extra carbon in. These have also been shown throughout decades of medicinal chemistry to cut down on proteolytic susceptibility of peptides. And then in some position, but not all positions, you can put in D amino acids. So then the big question is, of course, okay, you can put this all in. These were all in test peptides. What happens if you put those into the natural product? And so this is just showing by mass spec. In blue, you see the peptide, the MALDI mass spectrum of the starting material with different non-natural amino acids incorporated. And then in red, you see the product after you put in the enzyme. So the 4 dehydrations have taken place, and the 3 cyclizations have taken place. And you can now remove the leader peptide and look for the activity then of the final product. So when we first did this in 2009, we fairly randomly made a panel of, I think it was 12 analogs. So in the bubbles, you see the natural structure, the natural amino acids. You see the 3 rings that are present in lactosin 481. And then in the arrows, you see what unnatural amino acids we put in. And again, we just put them in at positions where we knew it was tolerated, but we had no specific rationale for what we were doing. We were therefore rather surprised to find out that already 3 of these molecules appeared to be more active than the natural counterpart. And so if you change this phenylalanine with a homophenylalanine, so you put in an extra carbon, something you cannot do by any post-translational modification process that I can think of, or you take the indole of a tryptophan and you replace that with a naphthalene, or you take the imidazole of a histidine and you replace that with a pyridine. These are 96 well plates. These are the wells. They are in agar, and we put in the wells our material. They are then going to diffuse into the agar. And so where it is opaque is where this indicator strain is still living. But while the molecule diffuses out of the well, where you see this dark circle is where you kill the bacterium. And so bigger circle means more potent molecule. And then we follow up in liquid broth with minimal inhibitory concentration determinations. It turns out that actually in one case this is a false positive, but in 2 of the 3 cases these are really more potent molecules. And so then the question becomes, what did you do? So you could have made a molecule that hangs around longer, that is not broken down. You could have made a molecule that maybe is taken up better by the indicator strain, or you could have made a molecule that interacts better with the target. And in 2009, we did not know what the target was of lactosen 481. So I'm going to skip 2 studies that we published where we looked at the mode of action of lactosen 481 that led us to believe that it was inhibiting cell wall biosynthesis. So that's that jump then. And so we thought that this particular step in cell wall biosynthesis was inhibited. So this is the so-called transglycosylation reaction. This is a reaction that is carried out by the PBPs, the penicillin binding proteins. And it's essentially a polymerization where a advanced precursor called lipid 2 is polymerized. So this has 2 sugars, 5 amino acids, and it's held by a greasy chain into the membrane. And this enzyme does a transglycosylation here, such that we add 2 sugars at each step to this growing polymer. And then later on, 2 polymers are going to get cross-linked, and that's essentially the peptidoglycan of all bacteria. So this is a good step to inhibit, and of course many current antibiotics that we take inhibit cell wall biosynthesis. And so Trent Oman went to Suzanne Walker's lab. She is at Harvard Medical School. She has this enzyme reconstituted in vitro, and we wanted to see is indeed the data that we think from doing experiments on organisms, is it indeed this particular step that we are inhibiting? And Trent was able to show that that's indeed the case. In a nice dose-dependent manner, you can completely shut this down. But if you look at the concentrations of peptides that we are using here, that's a little disappointing because that's, micromolar, and micromolar is good but not great. And the reason that this is micromolar, whereas the molecules actually have nanomolar activity, is because this is a class of molecules that turns out to not bind to the enzyme, but it binds to the substrate. And so that means that if, in this case, we have 4 micromolar substrate in this assay, we have to put in at least 4 micromolar of our compound to get complete inhibition. You might have nanomolar amounts of the enzyme, but that doesn't really matter. And so this compound can be more active as an antibiotic than as an enzyme inhibitor, because the amount of lipid 2 that is present on the cell surface of a microbe is much, much smaller than 4 micromolar. And we can deduce from kinetics that the affinities, and we don't have exact numbers, but that the affinities of these analogs for lipid 2 is in the nanomolar range. So these are what we think, well actually we don't know the stoichiometry in this particular case of lactosen 481 binding to lipid 2. What is really important for us is that we see very nice correlations between the killing activity and the enzyme inhibition activity. So molecules that are worse, which are really easy to make, these molecules also are worse in killing. But those few molecules that we have made that are better in killing are also better in inhibiting this protein, this enzymatic assay. So that's telling us that the improvements that we have made by, for instance, putting in, you probably cannot read this, by putting in a pyridine group, that improvement is because the molecule binds more tightly to its target. So even though we didn't know that at the beginning when we made these molecules, we're doing essentially what nature does too, which is further tinkering with the structure to expand the chemical space that we are interrogating past the 20 natural amino acids, to further fill in these little cavities that there might be that might lead to even improved binding to the target. Now, many of you are engineers. And so practicality is just as important as being able to do things. And everything I've shown you thus far is not particularly practical. We need to purify an enzyme. We need to click a leader peptide to a synthetic core peptide. And so scaling that up is not going to be very cheap. Actually, it's going to be really expensive. So we've known this from the start. And so we've known from the start that we would have to get a better platform to make these molecules after we get to better ones. So I'll tell you 2 improvements of the last year and a half or so that I think gets us partly there, not quite completely. And so in order to explain those improvements, I first need to tell you a little bit more about that leader peptide. I'll be honest, we don't know much about how this leader peptide works. We don't have any co-crystal structures, unfortunately, and it's not for lack of trying by Satish Nair's group. But what we do know is that you don't need to have these covalently linked. So if you take the leader peptide and the core peptide and you make these separately, you throw these in together with the enzyme, you do see activity, but you take a big hit. So the activity is much, much worse. You actually never get the final natural product out, but you get some activity, and that was kind of like interesting. Then Matt Levengood, who did this work also, took it one step further and showed that if you have no leader peptide at all, you take a further hit, but you still have a little bit of activity. You get a little bit of 2 dehydrations, no biologically active product. And so what that told us, that a model that we and others have favored, where the leader peptide induces a conformational change to turn on the enzyme, cannot be true. There must already be active enzyme present without any leader peptide there. And so our current hypothesis, which actually coincidentally fits very well with what most enzymologists are starting to think about how all enzymes work, how allosteric effectors work, how kinases work, is that there is always already a little bit of an active enzyme present. And in the case of kinases, you can see the 3% or so of the active enzyme by NMR with modern NMR methods. We haven't done anything like that. And that it's substrate binding or allosteric effector binding, or in our case leader peptide binding, that changes this equilibrium and pulls the enzyme completely to the active form, and then you can nicely modify your core peptide. So if this model is true, then there is a way that perhaps we might be able to no longer have to attach that leader peptide. So we got this really poor activity, but maybe we can get better activity either by throwing in a lot of leader peptide, and actually Pat Murr, who is here, has demonstrated that that is correct. Alternatively, and a better strategy, again developed by both Pat and Trent Oman, is to attach the leader peptide to the enzyme. Because you can do that at the DNA level. And so you can just have E. coli make you something that is, a blob, we don't know the structure, but where we have fused our leader peptide to our enzyme nucleotide sequence, such that in E. coli we express something that has the leader peptide already there. Now we had to by trial and error, change this linker. And so it turns out that a glycine-serine repeat 15 times does what we want it to do. So now this enzyme can take a core peptide and turns it into a final product without any exogenously added leader peptide. So our constitutively active fusion protein, or confusion protein as we like to call it, is active. It comes out of E. coli active, and we can give a core peptide. How general this is remains to be seen. We have 3 or 4 enzymes that all have in-trans activity, but whether this fusion will also work for these enzymes that have in-trans activity remains to be seen. But for now, this is really nice, because now we can just synthesize this. We don't have to put in these other steps of attaching a leader peptide and then later on cleaving it back off, which both, quite frankly, are low-yielding procedures. We can take a core peptide and we can then make analogs. So that makes that part, the discovery part, better. But scaling up is still as impractical as it was before, because you still have to purify enzymes from E. coli, you still have to synthesize a core peptide. So eventually you have to go back to fermentation. If you ask how are natural products that are currently on the market, how do you get those? It's almost always fermentation in the producing organism. And so how can we, if we find lactosin 481 analogs that are better, that have unnatural amino acids in them, how can we make those in an organism? And so for that, we first had to make these molecules in an organism that is tractable for these kind of applications, and that would, of course, be E. coli. And so, for reasons I'm happy to discuss afterwards, we had convinced ourselves that this couldn't be done. But then, as these 4 students have shown over the last year and a half or so, this is actually very readily done. All you need to do is co-express a his-tag precursor peptide with your biosynthetic enzymes from vectors that are commercially available that allow you to co-express. Purify it then with metal affinity chromatography, and then remove the leader peptide with a commercial protease. And that sounds so easy that you might wonder, why didn't we do this 10 years ago? Well, there's lots of reasons why we didn't do this 10 years ago, but I won't, in the interest of time, say that now. If you want to ask me, I'm happy to tell you. But we have done this now for prochlorosin, I'll tell you some more about prochlorogens in the next few slides. Actagardin, that molecule that is under development at Novector Biosystems. We've done it for niacin, we've done it for cinnamycin, which is a close analog of duramycin, the molecule that is in clinical trials against cystic fibrosis. So this works really well. And once we knew that this worked well, we could then see if we could bring unnatural amino acids in E. coli, because that has been developed, most notably by Peter Schultz, but also others have contributed. And what you do in the method that works best is that, again, you co-express, but in the gene for your precursor peptide, you put in amber stop codons that E. coli doesn't really use at positions where you want to bring in unnatural amino acids. And then Schultz has has evolved, I should perhaps say more accurately, orthogonal tRNA synthetase pairs that can put non-natural amino acids onto tRNAs in E. coli, and the ribosome, it turns out, this has been shown for more than 100 non-natural amino acids, the ribosome more often than not will incorporate those into your peptides. And so we are still at the beginning here, and so Nancy, she has shown that, for instance, you can put in para-benzoyl phenylalanine at different positions of this prochlorogen 3.3. And we did that because this is a photoactive cross-linking amino acid, and we don't know what prochlorogen 3.3 does. And so the goal here was to see if we could photo-cross-link this molecule with its target. So I'm going to tell you a little bit more about these prochlorogens, because I think it's an interesting story and it takes our bioengineering into a whole different direction. So we discovered these prochlorogens after we started wondering, why does nature only make one compound per bacterial strain? So by then we had demonstrated that we can put non-antibiotic peptides behind the leader peptide. Those are dehydrated and cyclized. So in principle, you could have expected that maybe an organism would make multiple compounds. You have the machinery there, so just put different peptides behind the leader. And we could never see that. But as more and more genomes came out, at some point, Bo Lee, then a student in the lab, came to me and said, I think we now have what we have been wondering about. This Prochlorococcus has a very small genome. It has only a single gene for a antibiotic synthetase, so a dehydratase and cyclase. But it has 29 little genes spread out over the genome, although 7 of them are relatively close to the putative antibiotic synthetase. And so if you look at those little red genes, what they code for, and I should point out this was in collaboration with Penny Chisholm at MIT, who is the discoverer of Prochlorococcus. So we contacted her. It's a marine cyanobacterium. It's very hard to grow, so she grows it for us. It takes months to get a little bit of cell paste. And so you have to grow it in seawater. And so the leader peptides of those 29 little genes are all remarkably similar, or what we thought were the leader peptides. Then attached at the end of these leader peptides are the core peptides. And as I already alluded to, here we found what we were wondering why nature didn't do it. There's 29 different core peptides attached to these leader peptides, and there's not a single one that has homology with another one. So we have 29 different peptides. Collectively, there are serines and threonines at every position from position 1 to position 26. And there are cysteines at every position from position 1 to position 23. So by now, at the time, we had shown for 16 of these that these are substrates. I forget the exact count. We've done a number more. I don't think we've done all of them yet. But I do think that these are going to be all substrates. So I think that Nancy here, is it 25 that we have tried? Nancy's not here. I think it's about 25 that we have tried thus far. She is there. 27? 18? Okay. 18 that we have tried. That are substrates. And so for some of those, but not all of those, we have determined the structures. And again, we use this expression system in E. coli. And so Wei-Xing Tang by NMR has determined 4 of those. The other 2 that I'm showing you here are by MSMS. And when you look at these, it's really hard to imagine how this can happen. So how can one enzyme make all of these products, where in this case a cysteine attacks an alkene that is localized on the C-terminal side. This cysteine attacks an alkene that is located N-terminally. We have overlapping rings, we have rings within rings, we have rings that span 10 amino acids, we have rings that span 3 amino acids. So the way, as an enzymologist, I think of an active site is almost like a mold where something is set up such that it binds or enforces a certain structure of the substrate in order to facilitate catalysis. Well, it's really, really tough to think of an active site that can facilitate formation of all of these different rings. Furthermore, Wei-Xing showed that the stereochemistry of all of these are the same. So a model that we had initially where we thought, well, maybe the enzyme doesn't do so much, it does all the dehydrations. And then non-enzymatically these things cyclize. You really would have expected to not get all the same stereochemistry if that were the case. So how does this work? How is this enzyme able to make what we believe are 29 different natural products? And so we don't know, but this is our hypothesis. Our hypothesis is that actually this enzyme doesn't govern anything. That this enzyme only chemically activates the substrate, but that the substrate has inherently the information as to what rings are going to be formed. And that this enzyme has maintained its very low specificity during evolution because this organism for some reason wants to make 29 compounds. But that, for instance, the enzymes that make niacin have evolved to only make niacin and to make niacin really well by some evolutionary process. And so we've tried to test this, and so far the model seems to bear out. And so let me first tell you a little bit about the cyclization enzyme. So NISC we have a crystal structure for that was solved in Satish Nair's lab in biochemistry. So it has a zinc, 2 cysteines, a histidine. These are all fully conserved, and then there is an open coordination site that is occupied by a water in the crystal structure. There's a conserved histidine aspartate pair, and then there's a conserved tyrosine and arginine. In follow-up papers where we did mutagenesis, we know that these two are not important, but that these two and these three are absolutely required. And so the one thing that is kind of special about this PROC-M is that this histidine is actually also a cysteine. So PROC-M, this highly promiscuous enzyme that makes 29 different products, it has 3 cysteines, and that fits very nicely in the model that we have as to how this enzyme works. So nature, in many cases, farnesyltransferase, methionine synthase, and about a dozen other enzymes, use zinc for thiol alkylations by having the thiol bind to the zinc. That now allows you, it lowers the pKa, you can deproteinate it, and then it can attack an electrophile when it comes along. Now, the key is in all of these enzymes is that you have to have a net excess of negative charges. Because now this cysteine or this thiolate is electrostatically spring-loaded, if you wish, to go off the zinc and not stay at the Lewis acid, but go to an electrophile. So inorganic chemists have shown that the more thiolates you put on the zinc, the more reactive they become. And so in this PROC-M enzyme, this histidine is also a thiolate. So we have 4 thiolates, which model inorganic chemistry tells us give you the most active system. So we think that this enzyme can make 29 different peptides because it has souped up activity. And again, that the precursor determines what rings you're going to be forming. So the little bit of testing of this model that we've done thus far, the best one, there is a lot more in this paper by my postdoc Xi Zheng and current student EU, but the best one I think is as follows. They took the PROC-A leader peptide, so this is the leader peptide of this very promiscuous enzyme PROC-M, and they fused to it a completely different core that enzyme has never seen. So we're asking an enzyme that comes from a cyanobacterium that lives in the oceans to, in E. coli, make a molecule that is made by a lactic acid bacteria that lives in the udder of the cow. Or, structurally, we're asking an enzyme that makes 29 products, some of which we know and others we haven't determined the structures yet, to make this compound. And I'm showing you this particular one here because you see this cysteine dihydrobutyrene that is very similar to this cysteine dihydrobutyrene. PROC-M turns it into this product, but LCTM in that lactic acid bacterium turns it into this product. As you can see, this cysteine, rather than reacting there, which it does here, reacts here for lactose 481. So if this peptide determines where this is going to be reacting, you might think that PROC-M won't do what it does with this substrate, but that it will make lactosin 481 from this substrate. So we do this experiment and then do the test. As controls, we also have lactosin 481 that we get from the producing organism, and we also make lactosin 481 with LCTM in E. coli. And so the two controls are shown here, and then here's the experiment. And so this is a much larger zone of inhibition. This is actually the work that E did. It's much larger because we put down a lot more material. We were not expecting to get good activity. And so we further have to make sure, well, so what we don't know in this experiment, so we know we're making lactosin 481. What we don't know is whether we're making other stuff as well, and we need to take a look at that. My prediction is that we're making a number of different things, and that LCTM in its evolutionary history has evolved such that it's only making lactosin 481. But that PROC-M, again, because it wants to make 29 compounds for some reason, has not undergone that evolutionary process. But it clearly shows that the core peptide itself can lead to a bioactive product, and that the core peptide might be controlling that. We have one other piece of evidence, and that is for the cytolysin, a molecule that is made by Enterococcus faecalis. So let me first tell you a little bit about cytolysin, and then how this fit into this model that the substrate determines what happens, at least initially in evolution. So this is a proteinaceous substance that is made by Enterococcus faecalis. This is, of course, one of those organisms that was on that very first slide, VRE, vancomycin-resistant Enterococcus. This is one of the main causes of VRE. It makes a proteinaceous substance that has lytic activity against eukaryotic cells. In the literature, this is the only example where a lentipeptide kills eukaryotic cells, human cells, kills macrophages, kills neutrophils. Essentially is believed to turn off, or at least in part, your immune response. Despite the fact it consists of two peptides that both require, it's really interesting, so separately they have no activity, together they lyse cells. But despite its really long history, it was first reported, I think, in the 1930s, we don't know its structure. But the genetics, so since we now know the genomes of these Enterococcus faecalis strains, it suggests that these are lentipeptides. A few more things about cytolysin. If you take hospital-acquired VRE infections, they are highly enriched in cytolysin-producing strains, suggesting that these are virulence factors that are important in infection of our bodies. More alarmingly, that if you get infected by an Enterococcus faecalis strain that has a cytolysin-producing genes, you're fivefold more likely to die than if you are infected by a strain that cannot make cytolysin. These are only epidemiological data. We don't know what the correlation is. But still, we felt that this was kind of interesting. Let's find out what the structure is of this cytolysin and how it works. Because now that we have this E. coli system, Wei-Xing Tang did essentially what I told you before. So he co-expressed the two different peptides. In this case, there is one enzyme that makes both compounds. And so in E. coli, he then determined the structure. So this is the structure of the large subunit of cytolysin, hence the L. And this is the structure of the small subunit of cytolysin, hence the S. And so she showed then indeed that here is a control where we put down nisin, and again we're killing the producing or the indicator strain. If you put down just a large subunit here at this dot, you see nothing. If you put down just the small subunit, you see nothing. This is an interesting experiment. If you put a large subunit down here and a small subunit down there, they start diffusing towards each other. And so only in between, you see this zone of inhibition. So where they meet each other, they kill. But things that diffuse in this direction or in that direction don't kill because they're by themselves. And of course, if you put them down at the same spot, now you get this large zone of inhibition. So we're fairly certain that we're making the right molecule. We have not brought the Enterococcus faecalis into our lab that makes this molecule naturally. We have no interest in having that in the lab. And so from this data, we think that this is indeed the structure of the right cytolysin. Now we then did something that we've been doing the last 3 years or so, which is, quite frankly, it was more of a thing that a chemist does, which is to determine stereochemistry, than that we were expecting anything different from what has been seen before. So what Wei-Xing did, so just to remind you, this is what happens in all of the antibiotics that people have looked at. She hydrolyzed these in 6 molar HCl, derivatize the amino acids that come out of that, and shoots those on a chiral GC column. And much to our surprise, this molecule, in addition to having one so-called DL-lanthionine, that's this here, it also makes 2 rings that have different stereochemistry, this stereochemistry and that stereochemistry, which we call LL. So it's L here and L there. So one single enzyme makes one ring that has this stereochemistry, another ring that has that stereochemistry, and another ring that has that stereochemistry. Again, that doesn't fit to how we think about enzymes. Enzymes are stereospecific. They do things well. This enzyme does things very well too. It turns out that this ring is always DL, this ring is LL, and that ring is LL. If you look at the small subunit, you hydrolyze that and you do the same analysis, you find out that you have one LL ring that turns out to be this one, and one DL ring that turns out to be this one. So if you then look a little bit more closely at which ones are LL and which ones are DL, you find out that all of the ones that are LL have, when that cyclization takes place, 2 dihydroamino acids next to each other. So again, the enzyme doesn't control the stereochemistry. It's a substrate. Once you have 2 dihydroamino acids next to each other and you cyclize, now the stereochemistry is LL. When you don't have 2 dihydroamino acids next to one another, you get DL out. And to confirm that then, Wei-Xing looked in the literature to see if there was any other genome sequences or any antibiotic sequences that had that CXX dihydroamino acid motif. She found several, purified those, and showed that those also have LL stereochemistry. So we're pretty convinced that it's the substrate there that has determined that stereochemistry. So it's not proof, but I think fairly good support that this is what's happening. Okay, so let me summarize. So I hope that from here on you will remember the acronym RIPS, that these ribosomally synthesized natural products are much more widespread than anticipated, and that they're fantastic molecules for bioengineering. Not just the lantipeptides that we work on, but those other classes of molecules as well. Because this leader peptide directed biosynthesis is almost a godsend when you want to change things. And you can keep things that you later on are not going to get into your final product. Keep that the same and just change what you're going to get in your product. Natural combinatorial biosynthesis is found thus far in organisms with small genomes living in nutrient-poor conditions. And then these RIPs are really well suited also for genome mining, so you can find new examples. So I have tried to name the people that did all of the work, because of course I didn't do any of it. Just very briefly, Trent Oman is now at Dow AgriScience, Matt Levengood is now at Seattle Genetics, and Bo Lee is an assistant professor at UNC. These students are still in my lab, done really a phenomenal job in teasing out this story and also trying to see what we can do with these systems. Xingang Zhang was a postdoc who is now an associate professor at the Shanghai Institute of Organic Chemistry, and Qi Zhang is a postdoc that is still in my lab. And so funding from HHMI and NIH, great collaborators, Penny at MIT, her postdoc Daniel Scher, Suzanne Walker I mentioned at Harvard Medical School, and then Satish has been really great in solving crystal structures of some of the enzymes that we work on. So thanks again, thank you for the invitation, and I'll be happy to answer any questions.
Well, that's a very fascinating talk. Any questions?
I'm curious about some of the precursor peptides that you're finding aren't found in nature, but when you assemble them, they seem to be even more effective than nature seems to. Why hasn't nature why haven't these come out in the past is kind of what I'm wondering. Right?
Yeah.
Are they more fragile, or are they not as useful in some other ways that we haven't discussed?
Yeah, so I think there's 2 answers to that. So when the analogs were made by site-directed mutagenesis, I think probably the compounds that are better for our purposes are not necessarily better for the producing organism, right? Which lives in a niche, which doesn't care about the human pathogens. It cares about whatever is surrounding them. So that's one part of the answer. The other part of the answer, when we put in non-natural structures, the structures that we put in, I don't know of any chemical way that nature can put those in. I don't know how you can take the natural amino acids and use, unless you use non-ribosomal peptide synthesis, but by using ribosomal synthesis, biosynthesis, how you could put in a pyridine from the naturally occurring 20 amino acids. That would be a transformation that could be Nobel laureate worthy if you were able to take a phenylalanine and turn it into a pyridine. And I don't know how to do that. So I think there's two parts to the answer. So better molecules where we take the building blocks that nature makes is, I think, because we have different purposes. But if we use building blocks that I think nature cannot, and that's really why we like this, because this brings us into an area of chemical space that you otherwise couldn't access, that's the reason why you don't find those in nature. Yes, okay. I'm now under a volley for my own students.
So in talking about substrate control, cyclization reactions, how does that then change a potential approach to bioengineering? So if the substrate, the particular sequence of amino acids has a lot of information about how the rings are going to be made, we start changing some of those amino acids in bioengineering approaches, still relying on the enzymes to put in those modifications. Does that kind of give you a snag there, that there are only certain...
Oh, absolutely. So I would say from a just scientific perspective, this is all fascinating. But for bioengineering, it's not a message that I like that much, because it's saying that there are certain things that we will never be able to do unless we engineer the enzymes and the substrates, and that's now going to be probably pretty challenging. So I think the way to overcome that is that, as you know, there are hundreds of these in the genomes, right? So if you want to make a certain ring, so part of what we do is trying to make cyclic peptides for other purposes. If there's a certain ring that catches your attention, like the Dutch group that made enkephalin analogs, well, just look for enzymes that make a ring of that size, and then use that enzyme for your engineering. So I think there's a way around it. Because I think Proc-M has maintained this, we will make whatever the substrate wants. I think these enzymes that have evolved to make a single product, I think these have evolved to actually make what they want. So I think if you're an organism and at some point you stumbled across niacin, niacin turns out to have 3 modes of action. That's one of the reasons why resistance has been very slow to develop, because it's very difficult to have resistance against something that has 3 different modes of action. So the niacin producer has optimized its enzyme, or has during evolution, such that it makes niacin and makes it extremely well. Even though maybe initially niacin might have been something that was formed in 2%, but that gave an evolutionary advantage, and then evolution started, such that now it's the only thing that Niacin makes. So I think it does have some disadvantages, especially for things like a highly promiscuous enzyme, but that it can be overcome by just taking advantage of all that information that is out there.
I have 2 questions. So what's the rationale for increasing the stability as well as the potency of those small peptides?
Yeah, that's actually a great question. I mean, you could say niacin, used for 40 years, almost 50 years, nanomolar activity. Why would you engineer it, right? It has great activity, it seems to not induce resistance. The reason why we want to engineer it is because of the half-life in blood. So I would, even though it's great to use niacin as a food preservative, I think that 10 years from now, if the resistance problem increases as is predicted, I think we would all give up on niacin in our food if it helped us from a lethal infection. It would have to be administered by IV. So if we can overcome the proteolytic susceptibility that molecule has in human blood right now, its half-life is about 40 minutes. So there are, again, things that you can improve that are improvements specifically for human use, not necessarily for what the organism initially intended it for.
Is there any guideline to to increase the stability more rational instead of trying to
Mm-hmm.
library-based?
Yeah, so I think, if you know where the compound is being broken down, bringing non-natural structures, especially if it's proteolysis, bringing non-natural structures at that position, that's what the pharmaceutical companies have done for decades.
Okay.
So many of the peptide isosteres and peptide mimics that are current drugs, they will mess around with the part where normally you would get proteolysis, and they will put in some of the structures that I showed you. And so for that you do need to know what the vulnerabilities are of your molecule, and then you can change those.
Another question, do you think from your personal experience, what's the biggest challenge for the discovery of natural product?
I think, and I think some of my students will agree, I think it's actually purification.
Purification?
Yeah. No, purification of the any natural product.
Okay.
So in the 50s and the 60s, natural product discovery was such a big part of drug discovery that there were people that were just phenomenal in taking a very complex mixture and teasing out a molecule that is in there that might be present in 0.05%. With the pharmaceutical companies closing their programs down, with academic research not having supported natural product discovery at all, because it is inherently non-rational. It's essentially, taking plants, grinding them up and seeing if you find something, or taking samples from deep marine organisms, sponges, grinding them up and trying to find them. So study sections have not taken well to these discovery type programs, because it's like, well, I can give you $250,000 a year, but I have no idea what you're going to find, or I can give someone else $250,000 a year, and there at least I know what kind of is being investigated. And so that means in academia, natural product discovery groups are either close to retirement, the PIs, or just gone. And in industry, the people that knew how to purify these molecules are all retired, and no new influx has been. And hence, to a large extent, we don't really know very well anymore how to purify molecules. The state of the art is pretty much what it was 20, 30 years ago. And so I think that that's the biggest hurdle. Something very simple, analytical chemistry, to quickly, so even if we're able to, say, take 100 gene clusters and make all of the molecules that these gene clusters make in E. coli, to then actually purify them and structurally characterize them, I don't think there's anything that has improved over the last number of decades. NMR has gotten more sensitive, but figuring it out is still the same. And so I think that is those are major hurdles. Unfortunately, NIH is starting to recognize that. And we don't do any of that work, by the way. Well, we do a little bit, but we're not specialists in that area.
Any more questions? All right, so let's take